Welcome, Amutha. Thank you so much for making some time for us today.
Thank you.
Yeah. So if you could just give like a brief background of where why you've been working with mitochondria and what you've been doing. Just to get to know you.
So I am currently the lead research scientist at Sense Research Foundation in the Mitotions program. I've been in the mitochondria field for a very long time. My postdoc was in mitochondrial biogenesis, and thereafter, in 2013, I joined Sense Research Foundation because I found the project very interesting.
And so what is the project?
Not many people know that there are two locations within each cell where you can find genetic material. One, everybody knows, this is the nucleus. That's what people think they inherit from their parents. But there is one other location in the cell where you find a tiny piece of DNA. This is in the organelles called mitochondria. And you inherit this exclusively from your mothers. So this small piece of DNA is about 16.5 kilobase pair in humans, and they are responsible for 13 important proteins. The genes for 13 important proteins are located within these this double stranded circular.
So each gene basically leads to one protein.
Yes. Right? It's a circular DNA, and the thing is that mitochondria have their own transcription and translation machinery within themselves. So they encode for 13 protein genes. They also have 2 rRNA genes and all the tRNA genes that would synthesize your proteins. But these 13 proteins, all the 13 proteins are part of the OXPHOS complexes. So that's one important function of mitochondria. There are several important functions of mitochondria, but 90% of your energy you get from these kidney-shaped organelles within each cell. So they perform vital functions.
Okay, and you've been looking into mutations in this area.
So there are several factors that predispose mitochondrial DNA to accumulate mutations. For one, they don't have any protective factors that are found in the nuclear DNA. The nuclear DNA has histones. Mitochondrial DNA doesn't have any of those factors. Number two, they because they perform this important function of aerobic respiration. The side products of aerobic respiration is reactive oxygen species and free radicals. These free radicals and reactive oxygen species are very harmful to make a DNA molecule. So that's number two. And number three, they also don't have very efficient repair mechanisms. So should there be some replicative errors, they don't have an efficient way to correct them. So all these factors predispose them to more mutation rate than the nuclear genome. So the project is that.
And what is the impact of a mutation?
These mutations lead to several mitochondrial myopathies. Some of them are from like point mutations to 1 or 2 of the genes. Sometimes there are deletion mutations of big chunks of DNA within the mitochondria. So there is there are different acronyms for these diseases. There are Leber's hereditary optic neuropathy, Leigh syndrome, Neela's, Merck, Keir-Sayre syndrome. They are all a result of mitochondrial DNA mutations. So
these are specific diseases.
These are
conditions.
Specific diseases because of mutations to the mitochondrial.
How about chronic?
Yes. So you could inherit mutations from your mother. So that's inherited mutations, but you could acquire these mutations de novo at any time during the lifespan.
Would that just happen at one point, or would it gradually happen?
No. So the thing is, it might happen at one point in time, but with age, the mutation could amplify, and it gets to a tipping point when the mutation load is such that it manifests in diseases.
So when you say load, does that mean in more mitochondria?
More mitochondria. So here is the other thing. There are only 2 copies for every gene in the nucleus. However, this small piece of DNA in mitochondria, there are about 5 to 10 copies of mitochondrial DNA per each mitochondria. And depending on the cell type, if the cell type has a high energy demand, then there are much more mitochondria in them. So like your heart muscle or skeletal muscle will have thousands of mitochondria in them, whereas resting cells would have 50 or 100. So if you multiply that and see 5 times per each, 5 to 10 times per mitochondria multiplied by several thousand mitochondria, so you have anywhere from 1,000 to 5,000 copies of mitochondrial DNA per cell. So 2 copies versus 5,000 copies. So when I mean a tipping point, let's say you acquire a mutation at some point in your life, and mitochondria. So in
just one mitochondria.
One mitochondria. But
and what could, like, just as a scenario, how could that happen?
So all these factors that I said before, free radical exposure, replicative errors, inefficient repair mechanisms, all of this contribute.
In infection, like are there any other things that can happen in your life that would trigger them?
Oxidative stress.
From an infection?
Yeah. That could be.
So it's mostly oxidative stress.
Yeah. So, and it keeps multiplying. They are going to divide. And there is sometimes certain mutations are selectively amplified. That it is also observed. So for a period of time, you wouldn't see any phenotype, but suddenly at age 40, somebody says their peripheral vision is getting lost. That's because, so not always that it's a mitochondrial mutation, but when it is a mitochondrial mutation, it is that this mutation has reached a certain level that you start seeing these defects.
Yeah, basically, so it takes over the mitochondria and it slowly spreads throughout the body. So somehow that one is prioritized versus the ones that are the originals.
Absolutely. Yeah, that's how they. The other thing is, it's hard to diagnose. Clinicians almost they want to eliminate everything else, and it's almost mitochondrial DNA mutation. It's underdiagnosed.
Right.
People just don't know they have till it's progressed so far.
So you gave a statistic earlier, 1 in 200.
Yeah, it's estimated that 1 in 200 humans carry some kind of a mitochondrial mutation in them.
Okay.
So
and as we age, more of us.
More and more, you keep accumulating these mutations.
Okay, but are there 60 people? I mean, people at age 60 with none, or is it, have you always got some? We're not really sure.
No, I'm not sure about that.
Right.
Yeah.
But the work at SENS research is basically it's based on the principle that there is going to be some oxidative damage to the mitochondria at some point.
Yes.
So that's
and the oxidatives you cannot separate the oxidative byproducts from the function of the organelle, right? It's meant to produce ATP, and these are the side reactions. The goal is to move these 13 genes to a location away from the assaults that they experience, put them in the nucleus, and try to express them at a functional level in the cytosol and get them to the right place in the OXPHOS complex. That's the
that's your work.
That's the Mitosense goal. Yeah.
Right. And so currently the project you spoke about today, was that part of that?
Yes, it is. We engineer the genes. We find optimal. It's an iterative process. We keep testing new features in the gene constructs, but what we do is every time we synthesize a version of any of the mtDNA genes for nuclear expression, we try to validate that in patient cell lines that we have, with specific mutations. So what we are trying to do is one gene at a time now. Of course, it will take a lot of engineering to accomplish.
So you were replacing mutations to see if you could get something that would work like the original? Was that?
No. So these mutations have happened de novo in patients. What we would do is we would take cells from these patients, and we would make laboratory models that we can culture them in the lab. So there is a distinct phenotype. Often these we work with cell lines that have one gene disrupted, a single gene disrupted. Sometimes it's more than one gene in the mitochondrial DNA, but we often
is this from the 13?
The 13. Yeah. So these patients, for some reason, acquired just a single mutation that disrupts one of these 13 genes. And as a result, because they are all part of the vital complexes 1 through 5 in the OXPHOS relay, they disrupt either assembly of the complexes or entirely the complexes are gone. So we test our engineered constructs in such clean systems. So we can say that if we are able to functionally express them from the cytosol and they go to the right location in the complexes, they should regain function. And that's the goal. Yeah.
Great. Well, thank you very much for your time today. Where could, I don't know if you do this, but is there anywhere people can reach out to you or find out more about your work? Is there somewhere you'd like them to go?
You can write to me at amutha.boominathan@sense.org. You can find my email address at the sense.org website, and I'll be happy to answer any questions.
Great. And you have a page on Sense of your research?
Yes. We have a Mitosense project page. So if you want to reach out, we have a public reach out portal as well. So you can submit your queries there, and it will get directed to me.
Great. Thank you very much.
Thank you.